Hey, Mrs. Paul here. I hope that you guys are doing really well while I'm gone. Mr. Kincaid is a great resource, and I am sure that he will do a great job teaching you guys and helping you guys out. But most of your teaching is still through these videos, and so I will continue to make these for you. And I thought that I would start by showing you some pictures of Kai. I sent out some remind text showing one picture, but I know that not everybody has remind and that a lot of you are still excited to see him. So here's baby Kai, and I went into labor early Thursday morning, and so I went to the hospital on Thursday, and he was born on Friday morning, and I had to have a C-section. So here's me and my husband, and here's when we first both met baby Kai. And then because I had to have a C-section, I was in the hospital until Sunday. And so here's baby Kai, like day 3. And then here he is going home. And he's a teeny tiny little baby. He's 6 pounds, 13 ounces, and was 18.5 inches long. So he's a little guy. And here he's wearing a 3-month size um, little sweater onesie. And it's just way too big for him. So he's tiny and super cute. So I will try to continue to show you guys pictures of baby Kai, and maybe even one of these videos I can record a little video of him so that you can see him too. So if you hear him during these videos, my husband is taking care of him, and right now he's kind of a little fussy because he's only 3 days old and he's trying to figure out the world still. So you might hear him. Anyways, we are continuing on in our unit on genetics, and so we are going to be learning about Mendelian genetics, and specifically today about the monohybrid cross. So before we get there, let's do some labeling in our notebook. Advanced class, here are the page numbers for you. Regular class, periods 4 through 7, here are the page numbers for you. This week our entry task is going to go on the first page listed here. On Friday you had a quiz, and after the quiz you were supposed to get the Gregor Mendel reading. If you have that finished, then it was a reading with some questions on the back to look at your own family genetics. If you have that finished, you can glue that in on page 140, so that's a page on the left-hand side. And then, well, for the regular class, page 140. The next page is for the questions about this lecture. So as you watch and listen to this lecture, there will be questions embedded. Remember, you are to write the questions in your notebook along with your answer, whether it's right or wrong. If it was a multiple choice question, then you can just cross your answer out and write the correct answer, but leave your old answer in there so that you can go back and see, oh yeah, I was wrong, but this is my thinking now. So that if you ever go back to your old thinking that was incorrect, you can go back and see, oh, this is why it was incorrect, and here's my new thinking. So after that, on the back of that page, you are going to be getting a practice page dealing with the genetics of SpongeBob. And then the next page is where you will copy the chart that will be at the end of this video. So if you haven't labeled these pages already and haven't glued in your entry task or the reading, if you can, go ahead and pause here and glue those things in, and then you can continue. So before we start learning about Mendelian genetics, we need to understand why it's called Mendelian genetics. So this is based off of Gregor Mendel. So you learned a little bit about him in the reading. It was a super short reading, but what you should have learned is that he lived during the 1800s. He was a monk who lived at a monastery in Austria, and at that monastery, everybody has their own individual jobs, and his job was to take care of the garden. And he was a geneticist. He had experimented with different animals before this point, and he was really interested in understanding the genetics of pea plants. And so in the garden, he grew some pea plants. Oh, here's a picture of him, so Austrian monk there. He grew some pea plants. And so There were some pea plants that were really, really tall, and there were some pea plants that were really, really short. And in order to be very specific about which plants bred with which plants, he used what's called cross-pollination. And he took the pollen from one pea plant, the flowers on it, and with a little paintbrush, and he transferred them to another flower on another pea plant and helped them do sexual reproduction through this way, so that specific plants were breeding with each other to create specific plants. Because what he wanted to do was take a tall plant and breed it with a short plant to see what the next generation was. So here are the parents and here are the offspring. So when he crossed the tall with the short, he found that both of the plants that resulted were tall. So he crossed those tall ones again together, and what he found was that in the second generation, the F2 generation, 3 out of 4 of them were tall, and one of them was really short. And so because of this, he was able to come up with some different laws. What you need to remember about genetics is that it is not always as simple as this. We can explain a lot of our genetics through the Mendelian methods, but there are some that we can't explain that way, and those are called non-Mendelian genetics. And so if you'd like to go above a level 3, then you can take a look at some different things with non-Mendelian, like sex-linked traits, or polygenic traits, and see what's going on there. Why isn't it as simple as what we're going to learn today? So one thing that he learned or taught us there was about a monohybrid cross, a single cross. So mono means one, and hybrid means a mix of different parts. And so here's the law of segregation. We recorded this already on a previous chart, our genes and alleles chart, I believe. Or no, it was crossing over chart, where in the end we get gametes that have separated the alleles to create gametes with half the DNA. So that each gamete has half the DNA and will combine with another gamete during fertilization to create a full set of DNA, a full cell. And so this was one of Mendel's laws, the law of segregation. So again, to segregate means to separate, and this law is again saying that the alleles in one cell separate so that now those new cells, those new gametes, one has one allele and one has another allele. So this is how it works in with the monohybrid cross. If our parents had the phenotypes of yellow and green, so these are pea plants, because remember, we're talking about Mendel, who experimented with pea plants. So the phenotype is yellow and green. The genotype for this yellow one is 2 capital Ys. So that tells us, because it's capital, that yellow is dominant and green is recessive, and so there are 2 little y's. The law of segregation says that it's going to split those alleles. So here's one allele for color and here's another allele for color. And it's going to split those into separate gametes so that there's one gamete that has one big Y, one gamete that has the other big Y, and one gamete that has a little y, and one gamete that has the other little y. So then when we create what's called a Punnett square, we're going to find what the next generation is. So I'm going to take these gametes and I'm going to place them either on the top or on the side here, separated. So here is the first gamete from the yellow pea, the second gamete. Down over here we have the first gamete and the second gamete. So notice that each box is a gamete that will create a new cell. And so each of these boxes is showing the offspring what they would have in their new cell. So when we cross these, I'm going to take this box and bring this Y down here, and I'm going to bring this Y over to this box. So this will now have the genotype that is heterozygous, capital Y, little y, And I'm going to do the same thing over here. Again, capital Y, little y. Down here, capital Y, little y. And then lastly, it is also capital Y, little y. So that the resulting phenotypes, if I take a look here, they're all the same. They're capital Y, little y. And so that will result in a phenotype that's yellow. And so then that means that my genotype, the only one possible, is heterozygous capital Y, little y, which means 4 out of 4 of my boxes were filled with that genotype, which means 100% of the offspring will be that genotype. So here is our practice. And so you're going to, or here is our chart. So this is the chart that you're going to record in your notebook, but I'm going to go ahead and go back and go to the next one and explain what your practice page with SpongeBob is, and then you can come back and record the chart. So here is our SpongeBob genetics. Here we got Patrick, and here's SpongeBob. And I'm looking at question number 2. In question number 2, you have to write the genotype for these phenotypes. The phenotype for each genotype. So here's a genotype, and you're going to write the phenotype. And so I went ahead and gave you these two. With a heterozygous genotype here, it says square shape is dominant to round. So because I have a dominant allele here, that means that this is square. And because I don't have any dominant genotypes here, that means that this is round because it's all recessive. Now I'm going to use this information for question number 4. And so this is about SpongeBob and Sponge Susie. And Sponge Susie is round, and SpongeBob is heterozygous for his square shape. So here is like on our previous slide, and I'm going to put SpongeBob's genotype along the top here. So I separated those alleles. And now here is Sponge Susie's alleles, her gametes. So now we're going to fill in each box here. So in the first box, it would be a resulting genotype that is heterozygous. In the next box over here, I'm going to cross this little s and this little s. So I get a recessive genotype. Down here, I have a capital and a lowercase. So that creates a heterozygous genotype. And then lastly, I have 2 little s's again. So question A asks to list the possible genotypes and phenotypes. So if I look here, I have one genotype that's the same. This genotype is the same, which means I have 2 possible genotypes. And I don't believe I typed it all in here, but to give you an example, here's one genotype, and I'm going to write what its phenotype is, which is square. So then you're going to go ahead and write the other genotype and phenotype. So the chances of a child with square shape, we're just going to count how many that is. And so with the square shape, I want the capital one, so I have 1, 2, out of a total of 4 boxes, which means that there's a 50% chance that they could have a square child or a round child. And so down here, the same thing with round, there's 2 out of 4, which is 50%. So you are going to go ahead and go back and record the chart, and then you are going to continue working on SpongeBob. After this SpongeBob practice, you're going to do an activity called Create a Baby. And so I'll talk more about that with you later. Good luck!